Okay, so I'm Heather Whitson, and I co-lead the Duke-UNC ADRC along with Dr. Gwenn Garden, who will take over here in a moment. I am a professor of medicine at Duke, and Dr. Garden is professor and chair of neurology at UNC. So the goal for today, we have probably about 20-25 minutes worth of slides to present, and then we hope that if you have any particular questions or discussion, now is the time to kind of ask the directors things that you may want to know about the ADRC. The goal will be to just kind of give you an overview of what the Duke-UNC Alzheimer's Disease Research Center is and does, to describe some of the resources that we have available, the cohorts and the samples and specimens that we are curating. So please have your phones ready for QR codes, because there are a few QR code opportunities throughout this talk for tools that you can use and access. And then a lot of this is gearing toward a few special funding opportunities that are open now, and that we hope you'll hear about, sort of a resource or something that is available to you through the ADRC that would help you in securing funding from these opportunities. So just to kind of get everyone on the same page, and I will remind everyone that we're recording this talk, so you will have access to it later. But the NIH designates 35 Centers for Excellence in Alzheimer's Disease and Dementia Research. This slide shows the states where those are currently located. Tennessee is currently a developmental center, and the rest of the states that you see have at least one fully funded Alzheimer's disease center. Now, Duke and UNC were awarded our center in September of 2021. So we're not the youngest, but we are one of the youngest of these NIH-funded centers. So we've been in operation for about three and a half or so years now. All right, so this is our organizational chart. This shows you the people who lead the various cores of the center. These cores are mandated by the NIH. So the NIH is pretty prescriptive about how these centers run. Each one of them has to run the cores that you see listed here. And but as you'll see in a moment, we do have a lot of flexibility about the theme and the particular activities that we do under these cores. In addition to Duke and UNC partnering together for this center, we also partner with several other centers and several other schools in our catchment area of Eastern North Carolina. So our partners include ECU, or Eastern Carolina University, UNC Pembroke at Pembroke, and NCCU, which is here in Durham. We have scholars and faculty members from each of those institutions, as well as Duke and UNC. So our theme here that we sort of chose to focus on is this idea of age-related factors across the lifespan that contribute to the development or the progression or the experience of Alzheimer's disease. So as we all know, and some of the most prominent genetic contributors to Alzheimer's disease, the link was discovered here at Duke by Dr. Alan Rosas and team. So we all know that a large part of Alzheimer's disease is a genetic disease, and that you inherit that genetic propensity at birth. But interestingly, unlike a lot of genetic diseases, people get 6, 7, or 8 decades of life before the disease manifests. And so our interest here is in trying to understand what are those sort of factors that evolve across the lifespan. So we're interested both in exposures across the lifespan that may determine risk of Alzheimer's, but also the biology of aging itself, age-related factors that are necessary to initiate the pathology of Alzheimer's disease, as well as the kinds of comorbidities or other exposures that influence a person's experience of living with dementia once they have the pathological features of Alzheimer's disease, which are amyloid and tau. So the problem with the existing evidence base is that there's sort of a gap when it comes to trying to study this critical part of the disease process that happens before symptoms develop. So most of the prior AD studies have either focused on older adults, they've enrolled older adults that already had symptoms and their age-matched healthy control peers, or they focused on these kind of high-risk, rare variant families where everybody in the family develops Alzheimer's pathology and symptoms earlier in life. And they tend to skew towards highly educated, high socioeconomic status, mostly European ancestry cohorts. And that doesn't really lend itself to understanding this kind of broader view of Alzheimer's disease as a condition that develops for decades before the memory loss is present, nor does it help us really understand all of the various exposures and lifestyles and comorbidity patterns that contribute to the disease. So what we're really trying to do is to fill some of those evidence gaps and also to sort of function for Eastern North Carolina as what we consider your friendly neighborhood concierge for Alzheimer's disease research that you may want to do. So a good way to think of us is we put on a lot of symposia, talks. If you go to one of those talks, a postdoc and her mentor walked out of such a talk and say they have a new idea about Alzheimer's disease, well, we, the ADRC, wants to be here to help. So perhaps what you would need to take action on that idea would be access to people who are interested in being in a study. Perhaps you need access to stored blood or cerebrospinal fluid or brain specimens from people who are well characterized with regard to their disease. Perhaps you need to be connected to experts or potential investigators. Perhaps you need didactic training or mentoring related to Alzheimer's disease and related dementias. Perhaps you need access to novel biomarkers, people with the expertise to tell you where you can have these biomarkers run and what novel biomarkers are available to you. Perhaps you need access to data sets and then the analytical expertise to be able to develop your methods and power calculations. Or perhaps you need pilot funding. So what we're trying to do is sort of build the resources that would give you access to all those things. So the first thing I'm going to tell you about is our memory and aging study. So again, all of those 35 centers that are funded by the federal government are required to run a longitudinal study that is relevant to Alzheimer's disease. The centers have a fair amount of leeway in exactly how many people they enroll and what kinds of people they enroll, and to some degree, what kinds of data they collect on these people. So our goal is to create a cohort of 420 people. These people have to be followed at least annually. And our goal is to deeply phenotype them with regard to their brain health and their risk factors for cognitive trajectories as they age. In addition, because we're focused on that period of time before most people are symptomatic with Alzheimer's disease, but where we hypothesize the biological changes associated with Alzheimer's disease are already happening, we start following people annually at age 45. So our cohort is people ages 45 to 80 at the time of enrollment, and they are followed annually. We've also set for ourselves the following metrics: that we want at least 20% of this cohort to be people from underrepresented racial and ethnic groups. And we want at least 30% of the cohort to be people who reside in rural census blocks. Up to 100 of these 420 people can be enrolled with mild cognitive impairment or early Alzheimer's, but the others we expect to be cognitively normal at the time that they enter the study. Now, we do want most of them to have a family history of dementia or a known ApoE4 gene. This means that this is a high risk, sort of enriched for AD risk cohort, so that we expect over time as we follow these people, many of them will transition to biomarker positivity, meaning if they don't have they don't have amyloid present at the time that they come in, they'll develop amyloid during the time that we're following them, and then ultimately develop cognitive symptoms and dementia. The things that we collect, some of these things are mandated. So there's something called the uniform data set that we're all required to collect, and we do. But in addition to that, we bank blood every year when these participants come in. We also perform some additional neurocognitive testing. The neurological exam and most of the health surveys are the required ones, but we also collect retinal images, a very detailed MRI protocol, And then every participant gets either cerebrospinal fluid collected and banked or a PET scan to determine their amyloid and tau status. We also do a whole battery of sensory and mobility measures. So we check their gait speed, their grip strength, as well as measure vision, measure hearing with audiometry, which is what you see being done here by Brittany, one of our amazing clinical research coordinators. And then olfactory testing as well. And all of them have an ApoE status determined. So we also have what we refer to as our young cohort. Now, this was an innovative idea and is funded separately from the federal money. But the goal here is that we want to enroll up to 100 people who are very young. These people are between ages 25 and 44, but they still have a family history of Alzheimer's disease or dementia. They get all the same testing that the regular cohort gets. But because they're so young, we don't follow them every single year. But when they turn 45, they can join the MAS cohort and begin to be followed annually at that point. But still, the one-time data collection and banking of CSF and plasma on this group of people provides this kind of really rich resource for, number one, if we begin to see signatures or patterns in the biomarkers that look like they occur and the transition points between, say, right before a person develops amyloid or right before a person develops tau or cognitive symptoms, we can sort of benchmark them against this younger group and make sure that what we're seeing is truly a disease-related pattern and not just an age-related pattern. Also, this group is really useful for biomarker development. If we have an innovative biomarker that we develop and that we identify in people who already have disease or who have biomarker positivity defined as amyloid positivity, we can go back in this young group and see if we can see differences in this biomarker evolving even at these very young ages, and perhaps differences in people that have an ApoE4 gene or don't, for example. So in terms of our longitudinal cohort, that main cohort where our goal is to have 420 people at this point, and again, we're in the 4th year, so we have about a year and a half of data collection left to go before our first 5-year funding cycle is over. We currently, as of a couple days ago when I checked the website, we had 298 people enrolled in the longitudinal cohort. 281 of those are active, meaning that some of them are lost to death or follow-up or withdrawal at this time. But you can see we're also doing pretty well with regard to the metrics that we set for ourselves. We have over 20% of the cohort that I identify as something other than white European ancestry. We do have more females than males. We are always looking for more men who want to be in the cohort, but the high number of females also helps us with our perimenopausal subset. So women who come into this cohort who are ages 45 to 60 at the time they come into the study, and currently we have 90 of those, they get extra blood tests and questionnaires to sort of characterize where they are with regard to menopause, because that is one of those transition periods, one of those age-related factors that has been linked to Alzheimer's disease risk. We currently have almost 30% of our participants who reside in a rural census block, and over half carry an ApoE4 gene. So I'm going to turn it over now to Dr. Garden to walk you through the rest of the slides.
So I am lucky enough to be able to show you some data, some of the initial data from this cohort that we've collected thus far. So these are examples of biomarkers that we collect on the individuals in the cohort. And so we'll start with, I can never remember if people can actually see my pointer on Zoom. We'll start with the one that has the 3 axes, the A-beta 42/40 ratio, A-beta 42, and p-tau 181. And these are CSF biomarkers of Alzheimer's progression. And you can see that amongst our cohort, which is predominantly asymptomatic individuals, you'd see both a correlation that you would expect, where you have the lower A-beta 42, lower ratio, and higher p-tau 181, in a subset of individuals that correlate with each other. And and are representative of those who are already demonstrating Alzheimer's pathology. And so that subset of the cohort, some of whom may or may not have elevated total tau, which is represented by the color lookup table there, are the those who, if you were looking for samples from individuals with AD pathology, who we might be able to provide samples from those individuals or other types of data from those individuals. We also collect neurocognitive data and sensory function, hearing, vision, etc., smell. We already heard about that. And so, and we have good evidence of a correlation that executive function declines in a manner that's correlated with the declines in sensory function. And on the top corner, you can see that there's also correlations between changes in optical coherence tomography and the p-tau 181 biomarker. So it's been gratifying at this particular stage of accumulating this cohort to see that we're getting a nice correlation between those things that we expected in the cohort, even though we took a slightly different tack for recruiting so many of our cohort without symptoms. To be able to see these correlations are, how should I say, supporting some of the hypotheses that we had when we started. And next slide. So one of the things that's important for investigators, however early stage they may be, to know is that all of this data that we've collected, and this includes the biomarkers that I just showed in the last slide, but also MRI scans and additional biomarkers, additional neurocognitive assessments. All of this data is available for investigators to use in their research, and samples are sometimes available depending whether you're looking for serum or spinal fluid as well. And the question is, how would you get access to this data? Well, the place to start is a place we call the arena. And if you're thinking you might be looking for some of this data for your research or for samples, I would encourage you to get the QR code here and explore our location of all of this data in the arena. And there's a portal for you to get access to information about this data in something we call the gateway. And there are really two versions of the gateway, one which has a lot of demographic information and data about samples and MRI scans, and another which is all about something we call the brain bank, and it relates their neuropathologic diagnosis of individuals who had donated their brain to other factors that were collected prior to them donating their brain tissues. So those are really two separate portals that you can look at: the neuropathology portal for the brain bank, and then the gateway for the arena, which is all the data that we've been collecting in the ADRC since we started. Next slide. And so if you're interested in getting access to this data or these samples, we're going to ask that you submit your request through our resource request portal. And again, that can be found at this with the link in this QR code. And what do you need to use this portal for? Well, really, anytime you want to ask us for something, if you don't want to, you don't need to make an appointment with either Heather or myself. You could, but you don't need to make an appointment with a core leader. You can simply place a request in this portal, describe your experiment that you want to do, what your research is, where the funding is from, and state what you need. Do you need a letter of support for a grant application? Do you need data or images? Do you need biospecimens or biofluids? Do you need help with recruiting individuals? Or do you need someone to come to your group and speak about Alzheimer's disease? Or do you need a consultation with one of our core faculty about any of these biomarker type of assessments, or even we have biostatistics consulting for AD relevant research. So just to kind of summarize, what we have available, there are over 1,300 brains in the brain bank. We collect blood, DNA. We have some information from the samples already in terms of the A beta 40, 42, p-tau 181, total tau, a glial fibrillary acidic protein, and neurofilament light chain. But we also can provide you with samples if you want to look at your own a molecular marker. We are developing a roster of individuals who are interested in participating in research, but who are not part of our longitudinal cohort. So if you're looking for people who have never had any of this analysis done before, or if you're looking for people who are, a larger group of individuals with more severe dementia, or you're looking for caregivers, we can provide access to individuals on that roster. I will say the roster is really just getting started and doesn't have a huge number of names, but we hope with time that it will have a large number of people, who are interested in participating in research. We can help with access to the North Carolina Registry for Brain Health, which also provides access to individuals who may be willing to participate in research. And we mentioned the statistical support and consultation, either for grant applications or for the evaluation of data that you may have, and the development of publications or manuscripts on that data. And then experts, collaborators, and other educational opportunities are another major offering of the center. And all of these resources are available either through that resource request portal or through contacting core leaders or other individuals who are working within the ADRC. We also offer resources to the non-academic community. There's the Outreach Core does a lot of work with the community on dementia awareness, connecting individuals with support services, and then we have, as you all know, an important component of our center, which is about workforce development. So we provide this forum, this ideas forum, and then the Slam Dunk, which is a trainee research symposium every year. And then Heather, you want to go to the next slide? And we also provide people with access to programs that are about career development for doing research in the Alzheimer's field. So these are two individuals who received awards and publications for their AD research through involvement in the center. This is all of us at the Alzheimer's conference in Philadelphia this past summer. Next slide. And then there's also the Alzheimer's Association has a new investigator award program. The letter of intent is due in a few weeks. This award is only available to investigators through working with an ADRC. So if you are interested in applying for this award, it's really primarily for late stage postdocs and a very early career faculty. So if you happen to be in that phase of your career and interested in applying for this, it is a very competitive process, but two individuals supported by our ADRC have been selected for this award in the last 2 years. So if you've got a great project and you would like to apply for this, you need a mentor who is affiliated with the ADRC, and then you can submit a letter of intent. Then the applications are due in January. Next slide. We also have other pilot award opportunities. This particular one listed here is about using blood-based biomarkers. If you have ideas, again, we have blood. And so it might be worth considering applying for this award, which the applications are due in January. And then the other information about how to contact us is all listed here. These are all on our website as well, about who to contact for what. as you all know, Jackie, who's managing this ideas forum, is a great contact initially, and she can help put you in touch with others. And you can also just submit general questions, especially about things like letters of support or how to use the resource portal, to that email below, the Duke UNC ADRC at duke.edu. We have a new website. We hope that it's going to be really helpful for investigators. You can access the arena, you can access our research portal, a library of resources, etc., all from this website very easily. And so please check that out. In addition, we'd ask for your help if you do receive any support, even if it's just a conversation with an expert or some statistical support. We hope that you will acknowledge our funding, this P30 AG 072958, in all of your publications that are derived from that support. And also, we want to hear about your publications. We have we do a lot of sending out information about the research people are doing through our newsletters, and post and news on our website regularly. So please tell us about your publications at this link. And these slides should be available on the website, as well as a recording of this presentation.
Okay, now I think we are open to questions. So we want to hear from you all, particularly if there's anyone who is planning to apply for the NAC New Investigator Award. I believe we have at least Juan J, I think I saw on the call, and he is one of the recipients. So you've got someone who has done that successfully here on the call, if anyone's interested in that. And maybe Mike DeVinny's on too, but I'm not sure. Giving people just a few moments in case anybody is putting questions in the chat.
Heather, do you want to just maybe pull up the website and show them what the resource portal looks like?
Yes, that was actually exactly what I was trying to do, but I'm fighting with I have like 3 monitors set up and fighting with each other right now. Share the website. All right, can people see that? When is the website sharing?
Yeah.
Okay. Okay, so as you can see up along the top there are navigation bars that you can go to various things, but you can also scroll down. We are doing a supply drive along with the Dementia Alliance of North Carolina to help folks in Western North Carolina. But then you can see we've tried to sort of organize things that study participants might be interested in, things that researchers might be interested in, and things for family and caregivers that visit our center. So under resources for investigators, that's where you're going to find the quick links to that resource request portal, where you can request things like a letter of support for your grant application, as well as it's where you can request any kind of sample, or if you need a speaker, or looking for an educational opportunity or anything else. You just sort of go into this request portal and then click what it is that you need, and it'll kind of walk you through the process. This is also the Arena Gateway. So if anybody followed the Gateway QR portal through your phone, you might have seen that it doesn't come out super nice on an iPhone, but if you look at it on a laptop, like I'm doing, you get a nice visual of all of the data that we have available. And I'll show you in a minute how you can kind of do some like nice little searches. But this is showing the participants that we have collected data on through the ADRC. This is putting together both the young cohort. You can see here people ages 20 to 45. We have 41 of those, as I said, and the rest of them are those people in our longitudinal cohort. If you go down here, you can toggle to looking just at the neuropathology bank. That's the brain bank, and you can see what samples are available of brain tissue in that brain bank. There are actually even more samples than what are listed here on the website, but these are the ones that have been sort of modernized in terms of their characterization and pathology work. And then in addition, there's this is the biospecimens bank. This is a collection of data that is where what we have available is blood and CSF on people who were characterized in the clinics. So you can see that this includes a large number of clinic patients. So we don't have all the data on them that we collect on the research participants that come in and undergo the 3 days worth of MRIs and retinal images and sensory and olfactory testing and all that kind of stuff, but they do have a clinical diagnosis and banked blood or CSF. So still very useful. I'm going to check the chat because I see a few. That for the new investigator award program, applicants required to identify a mentor. And so the mentoring program actually is my understanding, and maybe there's someone on who participated in it last year, but I believe that they pair you, they kind of assign you a mentor from the national network of ADRCs that mostly just can kind of provide like career development advice, but then also will read your aims and research strategy and give you feedback. And in some cases, that person ends up being part of the proposed project, but they don't necessarily have to.
So, Heather, I think the last year, and I haven't looked at the RFA this year, but last year I had to, for all of the UNC applicants, I had to identify a UNC mentor. So I think there's like multi-layered mentorship involved, so that I had to document that they were affiliated with the ADRC and they had a UNC mentor.
Yeah, you have to say that they're affiliated with the UNC, and that is actually an important point, is that, so I think last year we did have about a dozen applications that went in from Duke and UNC, and Gwenn and I have to, so Gwenn and I are actually the PIs of record for these grants. So because you have to be affiliated with an ADRC in order to apply for one, it goes in with either Gwenn or I as the PI, and we put in a letter of support, basically vouching for the fact that you are, that we claim you're affiliated with the ADRC, and exactly as Gwenn's saying, that we sort of point to like a person within the ADRC that you're paired with. But then in addition to that, you get paired through to somebody who's at a different ADRC somewhere else in the country.
But in addition, I think if let me say this: if let's say your research, your main collaborator is a senior scientist at another institution, that person can also be a mentor, right? You just and they could maybe be the most important mentor. There's nothing to exclude that, but you do have to have a local ADRC affiliated mentor as well.
And they are planning this year, I understand, to give again at least 10 national awards. Like we said, these are pretty competitive. So a lot of the centers submit, multiple people affiliated with each of these 35 centers submits an application, and they end up funding between 10 and 12 now a year. I think actually The year that Mike DeVinny got it was the last year that they only gave 2. So Mike DeVinny got one when they were really, really hard to get. They're still, it's a competitive process, but we do know that we have a good chance because we've gotten. And I see a question here about, do you plan to release genetic genotyping or whole genome sequencing data for the participants? So our plan right now is that we, with our own budget, don't plan to do that. One thing that we will say as part of this is that if you come in and you request to use the data and say you've gotten a sponsored project or some kind of funding to be able to perform analyses on the data that we have, we do ask that after you do your study and publish what you want to publish, that the data that you collected comes back into the data set and is then available for other people in the future. Our integrated data set database, the ARENA, does have an Oracle background, and it's completely able, it was designed actually to be able to handle omic and genome data. So our data set can certainly handle that. And we hope over time that it will be included in the data set. Does that answer the question?
So one thing I want to add to that question, if someone is specifically interested in genetic resources for the study of Alzheimer's disease, there are important national resources. There's something called the Alzheimer's Disease Sequencing Project, and then there is genetic data in other parts of the ADRC ecosystem that incorporates data from other ADRC participants around the country. So lots of genotyping, array genotyping, and whole genome sequencing has been done on many individuals who have this unified data set, and you can get access to that data through those national resources. The ADSP is really mostly just GWAS, like it's just genetic data. And I mean, it's actually not just US, there's a lot of sequencing information, but they don't have the UDS on all the participants. But there is also a large amount of genetic data on people for whom there is a UDS. So if you have interest in accessing those resources, I would encourage you to reach out to me or Heather. We can, show you where to find those resources or recommend collaborators at either institution who are using those resources. But we are not generating sequencing or arrays on our participants. Now, we do have DNA, as Heather said, so if people want to do that type of work, they can. But we also send that DNA to central resources, and some of our participants may end up getting sequenced or have array genotyping done. The only thing that we have for every participant available through our resources locally is their ApoE4 genotype.
We also do have some amyloid and tau data available from the CSF, from those who have contributed CSF. We could tell you their amyloid and tau status. And you can see
and all the biomarkers that I mentioned. We should have data not on every participant because it's kind of slow to come through, but we do have that protein data. But I think Andrea was specifically asking about genome sequencing data, and that we are not getting.
So you can see here, I'm scrolling right now through, this is the that gateway to the ARENA. So this is showing you on and understand that there's always a bit of a lag of when a person gets enrolled and when all of their data becomes available, because it gets sent off and batched for assays and things like that. Usually some of the first data to come in are age and race. And so the numbers down here give you a good sense of kind of where the cohort stands. The ones in the middle sometimes take a little bit longer for those variables to be determined. or for that data to be collected. But you can go through and you can see, like, for example, if you were interested in the ApoE profile of the people that have thus far had their ApoE status determined, one thing that's really pretty striking is the fact that, as I said, over 50% of this cohort carries an ApoE4 gene. That's by design because we enriched them to be people with a family history or who had already knew from doing 23andMe or something like that they had an ApoE4 gene. But this is a great database to do comparisons of how other risk factors may evolve differently in middle-aged people with or without an ApoE4 gene. Sharing just so I can see if anybody wants to put a hand out.
Shazad, are you looking for the new investigator award? Like who has received them from the Alzheimer's Association? Is that the award list you're looking for?
That's what I'm looking for.
I think we got an email with that list, but I don't remember when. Oh, do you remember when?
I think I have it. I can forward it to you.
Okay. Yeah. Yeah, so I think there was 1 year with 2 people, and then there was this first year with 10. And so there's only 12 people on that list, but we'll send it to you.
Thank you.
I mean, I looked at it and my impression was it's a very wide variety of different types of research, everything from caregiving to the very basic science of, cell biology. And the thing that was common was, I would say, of the 10 that I looked at, like the vast majority were assistant professors. There was only a few people who were senior postdocs.
I think if we have no further questions, we can probably let people go and give people a little bit of time back.